Hello, everybody. This lecture is part of the OpenCAPI Big Data Analytics course, where we discuss genomics pipelines. In this lecture, we will discuss how we can scale up the Gatk genomics pipeline using big data analytics techniques to increase its parallelism and increase its execution efficiency and reduce its execution time. My name is Zaid Lars, and I'm from Delft University of Technology in the Netherlands. And this lecture is based on the work by Mushtaq et al. On Spark GA, where we use Spark to scale up the Gatk DNA analysis pipeline. As we discussed in an earlier lecture, the amount of data that is being generated in the genomics field is considered one of the biggest and fastest growing big data sets globally. At the same time, the analytics pipelines that are used to analyze this huge amounts of data is usually compute intensive, which eventually requires a lot of time to process the data that's collected. One of the most widely used pipelines in the field in order to do DNA analysis is called the Gatk pipeline, which consists of 5 different stages. It starts by a DNA mapping stage, which is usually done using BWA mapping algorithm. And then this is followed by a sorting stage where you sort the mapped DNA short reads according to their mapped locations, and then marking duplicates reads so that these duplicate reads are not used in downstream analysis. Then we use in-the realignment to be able to locally adjust the location, the map location of the reads to get a more accurate alignment. And then base recalibration is done to adjust the quality scores of different base pairs to improve the overall base quality score assignment. And finally, we end up with a haplotype caller to do variant calling on the reads and identify the mutations or the variants in the sample DNA. This is state-of-the-art pipeline that is widely used, and it requires a number of different tools to be used to be able to get the results, the variant call results. However, if we look at the CPU utilization of these different stages as they are executed on a compute system, we notice that many of these stages do not fully utilize the hardware available for the tools as they are executed. This chart shows an earlier version of Gatk with the x-axis showing the execution time and the y-axis showing the CPU utilization of the different cores on the node that the pipeline is being executed on. And we see that only very limited parts of the pipeline are actually able to fully utilize or utilize a big portion of the cores on the cluster, on the CPU nodes. Many parts of the time, the the CPU cores are actually idling, waiting for disk access or waiting for memory access to be able to continue their execution. So the pipeline does not efficiently utilize the actual available hardware infrastructure. And we need to improve this utilization efficiency to be able to get a better execution time. If we look at the pipeline and the type of data that the pipeline is processing, the data is basically genomics data, it's DNA data, and DNA is usually divided into into a number of pieces. Natural division of the DNA is usually can be aligned along the chromosomes, which are continuous strands of DNA that are present in most living organisms. In a human being, for example, we have 22 chromosomes plus a sex chromosome. And the idea is that we are able to process each one of these chromosomes independently from each other, so in parallel to each other. We also see that some chromosomes are bigger than others, so they contain more DNA data. And we can also subdivide the bigger chromosomes to smaller pieces and execute those also in parallel. There are a couple of existing methods to scale up GATK genomics pipeline in a cluster environment. GATK itself has GATK-Q that scales up the GATK pipeline in a supercomputing cluster. However, it's a hard to set up approach and it is rather disk intensive. There is Halvade, which is a big data scalability framework for the GATK pipeline. However, it's based mainly on the Hadoop file system, which makes it disk intensive and has limited use of memory and only allows for static load balancing. Our approach is based on Spark, Apache Spark, which is an in-memory big data analytics frameworks. It allows dynamic load balancing, which improves the parallelization capabilities and increases the efficiency of the analysis. And it effectively uses in-memory data and also has effective use of disk. And it's also rather easy to set up since Spark is becoming a standard in big data analytics. Our Spark implementation uses a pre-processing step that divides the input data set into a number of chunks. After that, these chunks are processed on the Apache Spark framework in 3 different stages. In the first stage, we perform the mapping algorithm of the short reads to a reference DNA, and we perform static load balancing on the mapped short reads. The second stage performs the sorting and also performs dynamic load balancing on the sorted reads. And finally, the third stage performs the Picard and the rest of the GATK pipeline stages. There are a couple of challenges in the implementation related to the large amount of memory used and also the parallelization and the efficient utilization of the processor and the CPU cores on the cluster. And this is what we'll address in the coming slides. The pipeline starts with a pre-processing step. The pre-processing first merges 2 FASTQ files, the left and right, into a single file, and then divides all the reads into chunks. This is the stage that we call chunk segmentation. The number of chunks that are created depends on the cluster size. The bigger the cluster, the more chunks we can create to enable the parallelization. This pre-processing step is performed only once before the data is processed. So the time taken to create these chunks is not included in the time calculations. After the pre-processing step, we move towards the mapping and static load balancing. All the chunks that have been created in the pre-processing step are now mapped using the BWMM algorithm, which can easily be scaled up to consume the available compute resources in the cluster. This way, we can parallelize the mapping stage to be able to run it on all different cores on the different server nodes. The output of the BWMM application is divided depending on the chromosomal regions where the reads are mapped to. This is called a static load balancing. Dividing the output of the BWMM reads depending on the chromosomal region allows us to reduce the memory needs when we want to perform the dynamic load balancing in the coming stage. The next stage is the sorting and dynamic load balancing stage. In this stage, files that belong to a specific chromosomal region generated by the different BWMN processes in the previous stage are combined together. This creates a file for each chromosomal region containing all the reads that belong to that region. And then these reads are sorted, and then the chromosomal region files are subdivided again depending on the actual number of reads in these files. Since the number of reads in each one of these chromosomal regions depends on the specific sample that has been sequenced, this number changes with every experiment. And dividing these files, depending on the number of reads in every experiment, ensures optimal load balancing for the specific sample. This is what we call dynamic load balancing, since it changes from one experiment to the other. The final stage is the Picard and GATK variant calling stage. In this stage, many mini GATK pipelines are created and executed on the different cluster nodes. Each one of these GATK pipelines analyzes one of the chromosomal region files that have been created in the previous stage. This enables utilizing the different nodes in the cluster and parallelizing the execution. The number of Spark containers that are created to be able to process and analyze the different region files depends on how efficiently we can use the cores in the cluster. Finally, each mini GATK pipeline generates its own mini VCF file, and all these VCF files are combined together into a one final VCF file for the whole analysis. In order to measure the efficiency of our pipeline and the scalability potential, we ran a number of experiments. Our experimental setup consists of 20 power 8 compute nodes, each containing 2 processors, each with 10 cores, capable of running up to 160 hardware threads per node. Each node contains 512 gigabytes of memory, and the nodes are connected using InfiniBand interconnects. The results show that our pipeline is able to much more effectively use the available compute resources on the cluster. This figure shows the amount of usage of the cores available on each node in the cluster, with the x-axis showing the compute time and the y-axis showing the utilization or usage, CPU usage. We see that the characteristics of utilization depends on the different stages in the pipeline. In the first stage, the mapping stage, and the third stage, the Picard and GATK stage, we see that the CPUs are very well utilized with very high efficiency. In the second stage, the sorting and load balancing stage, we see that there is significant amount of idle time, since there is a lot of communication taking place between the different nodes to communicate the data after the reads are mapped. When we look at the scalability potential of the different on a scalable number of nodes, we see that the first stage and the third stage can scale very well as we increase the number of nodes in the cluster. In this figure, we show a number of different cluster configurations going all the way from 8 worker nodes all the way to 19 worker nodes. And we see that the first stage and the third stage scale very well as the number of nodes in the cluster increases, while the second stage runtime does not decrease because of the large idle time and the communication cost. If we compare the utilization on many different nodes in the cluster, we see that there is uniform utilization of the different cluster nodes with very similar characteristics in terms of usage of the CPU on these different cluster nodes. We did an experiment and I tried to identify when we would be able to reduce the runtime of the pipeline all the way down to 1 hour. Based on the measurements we did until 20 cluster nodes and projecting this by reducing the runtime of stage 1 and stage 3 all the way down and keeping stage 2 runtime fixed, we see that we can bring the runtime execution time of the Gatk pipeline to under 1 hour when we run it on up to 35 nodes in the cluster. If we compare the accuracy of our pipeline with the accuracy of the baseline pipeline, we see that we have very high concordance and accuracy for our pipeline, achieving up to 99.1% of the baseline sequential Gatk pipeline execution. This ensures that the results that we generate from our pipeline are almost identical to the results generated by the baseline pipeline. This chart shows the cost estimation of running our Gatk pipeline on a cluster. Running Gatk on our Apache Spark solution allows us to have very efficient CPU utilization of the cluster, which means that we can run our solution at a low price. We saw that we can run our Gatk pipeline for under 60 minutes when we scale the cluster up to 35 nodes. The figure shows that as we reduce the runtime of the whole pipeline, we reduce the efficiency of our pipeline and increase the relative cost of running the pipeline on the cluster. This is a result of the inefficiency of running the stage 2 of the pipeline, since we cannot scale stage 2 down as we increase the number of nodes, which reduces the total efficiency of the pipeline. Translating this into costs, we see that we can run the whole pipeline for under $100 when we run it on a cloud service with power server nodes. The actual cost would be somewhere between $30 to $70 in a cloud provider such as SoftLayer. In conclusion, in this lecture, we showed that we are able to run the Gatk pipeline in the Apache Spark framework using big data analytics approaches. We were able to show very good scalability characteristics without sacrificing accuracy. The pipeline ran a whole genome sequencing data set of 400 gigabytes in under 1 hour on 35 nodes, and the cost of running such a pipeline was under $100 on a Power 8 cluster. This concludes this part of the lecture. Thank you very much for your attention.